So far, we've learned about causes of brain damage, neurological diseases associated with brain damage. We'll now be focusing on the neuroplastic responses of the brain damage. We'll be focusing on degeneration, regeneration, reorganization, and recovery of function. Neural degeneration, as you've learned in previous chapters, is a component of helping us smooth out our movements and our processes, and that is what would help in the development of the brain. But also, it is a component of disease, and that is basically the deterioration of the death of the cells. This, as it typically occurs, is a process that is not easily understood. We now understand that there is a lot of influence by the glial cells. And the reason why is because of the myelination that basically protects the axon. We look specifically on the cause of degeneration, and we find through laboratory studies that neural degeneration is often induced in a simple, controlled way. We could do this by cutting the axon, and that's what's known as axotomy. Now, there are two kinds of degeneration that would happen thereafter. You would have anterograde degeneration and retrograde degeneration. The axotomy that you could see here would be cutting the axon right at this area here. And based on what you would cut here, this would then bring about a response. Now, you could see with the anterograde degeneration, basically is the degeneration of the distal segment. That is the segment of the cut axon from the cut to the synaptic terminal. So you would see the anterograde actually moving, and you could see the dotted line right here just beginning to fade away. We would also see the retrograde degeneration, which would be that the damage or the degeneration would occur to the cell body. Notice how initially with anterograde it just started fading away here. Now you begin to see the process impacting the body of the cell in here, and that is the degeneration of the proximal segment. That is again the segment of a cut axon from the cut back to the cell body here. Overall, these could bring about also what's known as transneural degeneration, and that is when the neurons that were synapsed to each other in here now may degenerate because of the lack of connection in here. So please note that for transneural degeneration, in some cases it's called anterograde transneural degeneration, and that is because the degeneration would spread from the damaged neurons to the neurons on which they synapse, and in other cases it may spread from the damaged neurons to the neurons that synapse on them, and that's what we call retrograde transneural degeneration. It is also important for you to note that the anterograde degeneration, which would be here, this is a very quick process. Right after the cutting of the axon occurs, in about a few hours, you'd begin to see the neuron that would begin to get swollen, and within a few days, this would start breaking up into little fragments that would eventually disintegrate. The course of retrograde degeneration actually takes a little bit longer. In about 2 or 3 days, there would be some major changes that would be apparent in the cell bodies, and we find that those early cell body changes would either be degenerative or regenerative in nature. While neural degeneration is the deterioration of the death of cells, neural regeneration is the regrowth of damaged neurons. Please note that this does not appear to be as smooth a process in mammals and other higher vertebrates, but in some invertebrates and lower vertebrates, meaning reptiles and others, it does seem to regenerate at a faster process. You may remember the example of the frog visual system that was demonstrated or discussed in Chapter 9, and how Roger Sperry was able to rotate the optic nerve of the frogs. This was based on the understanding that the axons would regrow And we would see again that happening with frogs, but we would not be able to do that with humans. It is important to note, however, that we do have a capacity to regrow when we are in earlier developmental stages. However, once these neurons reach maturity, then regeneration is almost impossible in the CNS of adult mammals. And so when we look at the CNS, we know that it's a no-go. But if we look at the PNS, the peripheral nervous system, it may occur. And that's kind of like a 50-50 chance of this occurring. Here you can see a depiction of what would occur with the regenerational process at the PNS. Remember, it's not a guarantee that we would have regeneration in the PNS of mammals, but we could see in here that if we were to damage a nerve without severing the myelination component, meaning we would not crush or just smash the Schwann cell sheets. Keep in mind the Schwann cells is the white matter along the axon in the PNS. It would be called the oligodendrocytes if we were to be looking at these neurons in the CNS. But if we just damage the nerve and not the Schwann cells, then we could see that the individual axons would regenerate neatly across and to their correct targets. So we would damage the nerve here, leaving the Schwann cell intact. Regeneration occurs. However, if we damage the nerve and somehow we damage the Schwann cells, then these would be separated, and when they regenerate, they would not go back to the original target. They would in fact end up in incorrect sheets and reach also the incorrect cells. And you could see again how this would now regenerate to the blue, whereas it should have regenerated to the yellow. Now, when we damage the nerve and also we sever the ends of the Schwann cell sheets, and this is widely done at a massive level, then we run the risk of having these widely separated, and then we would not see any generation occur. They would just kind of be separated in there and crumble up in here and not regrow at all. You may remember in Chapter 9 we discussed as to how experience or the environment could help influence the organization of the brain in mammals. In this particular section of this module, we will learn on how the brain is also capable of reorganizing in response to damage to itself. Our understanding of the reorganization of the cerebral cortex because of damage has been limited to laboratory animals. We have focused and we've observed how the neural reorganization occurs after there is sensory and motor cortex damage. Now, sensory and motor cortex is ideal to study organization because of the topographic layout. You may remember that every sensory modality has a pathway that extends from in the hierarchy from the receptor end to the association cortex. So when we induce damage to the reorganization of the primary sensory and motor cortices, we have been able to study this under two major conditions, primary conditions that is. Following the damage to the peripheral nerves and also following the damage to the cortical areas themselves. Again, this consistent with the topographic layout. The demonstrations of cortical reorganization are about 2 decades old now, going on to 3. And there were 3 primary studies that were influential in our understanding of the way the cortex reorganizes after neural damage. In 1990, researcher Kaz and his colleagues assessed the effect of making a small lesion in one retina and removing the other retina. Several months after the retinal lesion was made, the primary visual cortex neurons that originally had receptive fields in the lesion area of the retina were found to have receptive fields in the area of the retina next to the lesion. This basically is something that occurred within minutes after the lesion. That is to say that whenever there was a damage to the brain compensated by organizing in a slightly similar yet different fashion. The previous studies were observed in mammals, but we did not know what organization, if any, would follow damage in humans. The demonstrations of the cortical reorganization and controlled experiments with non-humans provided some type of incentive to search some similar effects on human clinical populations. And so out of this line of research grew this particular interest to use brain imaging technology to study how the cerebral cortex of blind individuals change in response to damage. So the findings were consistent with a hypothesis that says that there is continuous competition for cortical space by these functional circuits. That is to say that without a visual input to the cortex, there is an expansion of the auditory or somatosensory cortex. And this would then allow for the individual to process information in a way that would be superior in the auditory sense now that the visual is lacking. Please note that there seems to be also a functional consequence of this reorganization, and that is that blind volunteers, again, have these skills that seem to be superior to those who are able to see. As impressive as this reorganization is in human adults, the recovery of the function after the central nervous system has been damaged is not very well understood. This is believed to be because of the improvements that are made, which tend to be modest or sometimes non-existent. Now, when we look at the individuals who were able to compensate for the lack of seeing in the case of individuals who were blind and who suddenly had a competition for the temporal lobe so that they could then hear better and process information better, this we do not know if it's something that is called a cognitive reserve. Now, a cognitive reserve is nothing more than the education and intelligence that would play a role in the recovery function. Individuals who would have some type of schema in their brain because of previous exposure would be able to come up with new ways to solve and accomplish a task, whatever it may be in question. And so we do not know if in fact it is a full reorganization and recovery of the function thereafter, or if it's simply just an adjustment to information that was already present in the cerebral cortex. Another possibility that can explain the recovery from brain damage is neurogenesis, and that is neurons that are developed in adulthood. When we look at laboratory animals, adult laboratory animals, there appears to be an increased migration of stem cells into nearby damaged areas. And some of these develop into neurons that can actually survive for several months. This finding has been replicated to a particular human who died 1 week after suffering a stroke. Although the stem cells can migrate short distances, there is no evidence that they can migrate from their sites of genesis in the hippocampus and eventually down to the areas where the damage occurred. And so in most cases we would say that although neurogenesis can contribute to recovery from brain damage, there is no convincing evidence that stem cells would contribute to the recovery. In this slide you can appreciate how when there is a perfect connection and the nerve has not been damaged, you have the area B of the skin that would synapse over to a cortical area that responds to touches at B. The skin area A, which would be at the bottom of the tip of the finger, we would see that it would synapse over to a corresponding area that touches at area A. Notice the size of area A and notice how this occupies a significant amount of space for B. However, if there were to be a damage to the nerve B, you could see how there would be a degeneration of this, and an almost instant reorganization would begin to occur. Whereas area A continues to be the same, this begins to kind of fall apart in here, and this begins to inhibit the responses at touch A. Notice how the expansion of area A continues to spread a little bit more to occupy that space that originally belonged to section B. About 6 months after the damage, you could see that the nerve B would have been completely eliminated, but now there would have been a collateral sprouting of some branches here, of axons, that would now occupy this area here. Notice how it went from being this small at the cortical area. Now, because of the absence of one, there is this competition for cortical space, and this would have allowed for establishment of new connections, even though we do not have the skin area B present anymore. In this last part of this module, we will be focusing on part of the reason why we're so excited about neuroplasticity. For the most part, our understanding of the brain should be used to develop treatment for humans who have sustained damage at the central nervous system. A lot of our research has focused on animals, and while that is beneficial, we know that it doesn't necessarily generalize or apply to human, specifically adults. So we'll be looking at how some of this research has run into some issues, but we'll also be left with a sense of hope as to the future of researching the recovery of the nervous system from brain damage. At the end of the 1980s, some investigators were interested in exploring the possibility of repairing damaged CNS tissue by implanting embryonic tissue. You may remember embryonic. It basically is at the prenatal stages of 2 to 8 weeks. And so what they wanted to do is they wanted to implant it near a damaged area in the brain, with the hope that the tissue would mature and replace all the damaged cells. Now, this gave way to a series of ups and downs in our excitement of this discovery. The first approach focused on individuals with Parkinson's disease, and the patients who had Parkinson's basically lacked dopamine-releasing cells, as you already know, and we were hoping that by transplanting the appropriate fetal tissue, the embryonic tissue, specifically to substantia nigra, we'd be able to improve their conditions. Initially, the signs were very, very positive. These individuals were able to accept the transplant, and sometime they were able to, we were able to see an alleviation from the movement and tremors that would characterize them. The effects of this in here, which were so favorable, were reported, and individuals prematurely began to offer this as a treatment at major hospitals. The results were very promising. There was a lot of excitement in this field, and we were able to see individuals just make some improvements. But unfortunately, this came to a halt when individuals about a year after the surgery began to have a lot of issues with chewing movements and some uncontrollable movements in the body. These results, although for a while they took us on a high of we were able to discover something, gave us and taught us two important lessons. One is that we should not jump into invasive treatments without understanding the dimensions of the effect that this would have. Implanting individuals with fetal tissue or embryonic tissue in the brain is very invasive, and if we do not fully understand this, we should not be doing that. Another thing that was discovered from this in here is that we should be a lot more careful at looking at the part of the brain, because even though substantia nigra may be involved with dopamine and at allowing us to have steady limbs and not have any tremors, we know that there are many other effects that this may control. And so implanting different stuff into different parts of the brain could bring about severe consequences. Thankfully, we've been able to recover from this not so pleasant part of our history, but as you know, through science, we unfortunately learn sometimes by making mistakes. And while some individuals fear that the consequences of the transplanting of fetal tissue would be long-lasting and it would deter from any future research of neurotransplantation, we now entered a very active stage and study of neurotransplantation. There have been hundreds of studies that have been conducted since the turn of the century, early 2000, and they've involved a lot of animal models through which we've been able to understand neurological disorders, and the tests are primarily done to benefit human patients, and we find that with stem cells and our understanding of stem cells, we've been able to successfully isolate from the human embryo and maintain in tissue culture. This because of the stem cells' ability to divide indefinitely, and because they're pluripotent, which is they're capable of developing into many, many different types of cells. Although not all pluripotent cells cannot develop into all cells, they can develop into several classes of adult cells that are needed. Per a study in 2002 by Kaz and colleagues, they were able to transplant stem cells into the spinal cord of a rat who had sustained some damage. And while the rat was unable to move prior to the the transplant of these stem cells, within 3 months they were able to see the rat gain mobility. While the improvement was not perfect and it never recovered the ability to move as it did prior to the injury, we did see that there was a lot more movement. And again, it's not a smooth movement, but it was noticeable as to the changes that were made after the transplant. The two previous methods were quite invasive, and they require a lot of training and a lot more research, as we do not fully understand things yet. But there are also ways in how we could promote recovery through rehabilitative training. Individuals that have suffered from strokes benefit significantly from training on a regular basis and from therapy. And this practice seems to substantially help the expansion of the cortical damage into the surrounding penumbra, and this would then allow the individual to recover as best they can. Please note that the recovery takes into account several other factors. If you are older when a stroke occurred, then the chances are you may not recover as well. Also, depending on where the damage was on a stroke, we may see different deficits. We know that with women, if they suffer from a stroke, they're less likely to lose their ability to speak, whereas with men, we're more likely to lose that. And also, the younger we are, the more likely we are to make a full recovery. Per a study with monkeys who had a stroke, we're able to see that they were able to recover hand function simply by doing rehabilitative training. The small ischemic lesions that basically brought about damage to the motor cortex of the monkey was treated by a program of hand training and practice. Basically, during 3 or 4 weeks, the monkeys would be asked to pluck a lot of little food pellets from different sizes, and the stimulation of the hands would basically bring about the expansion of the cortex that had been damaged. We also find that the principles that have emerged of studies of neurodevelopment is that there is this competitive situation, meaning that the neurons seem to be competing with other neurons for a lot of synaptic sites, and we find that those that do not, then they simply just fade away. That's what we call synaptic pruning. And so by constantly engaging in these rehabilitative processes, we are able to stimulate the neurons and strengthen those connections without being invasive at all. Because of this constant competition, one of the procedures to help individuals who had a stroke is called constraint-induced therapy. That basically consists of tying down the functioning arm for 2 weeks so that the affected arm would be receiving intensive training. Basically putting one out of commission because the neurons there are fine and there was no damage to that specific part of the brain. And what we find is that the performance of the affected arm typically improves over those 2 weeks, and there's also an increase in the area of motor cortex that controls that arm. Another rehabilitative training that is not invasive at all is what we call the facilitated walking. And it basically consists of helping individuals with spinal injuries that are not capable of walking anymore. And so what we do is we support them with a harness and we put them on a moving treadmill. The majority of their weight would be supported by the treadmill, but they'd be providing feedback as they learned gradually make walking movements. And as they do this in here, they seem to improve their ability to move their legs and their limbs as a whole. And the amount of support begins to be reduced when we notice improvement. There have been studies that have been done using this technique, and they find that more than 90% of the trained patients eventually become independent walkers compared to only 50% of those who received the conventional physiotherapy. So keep in mind that this is kind of like learning how to walk again for individuals who have sustained these injuries. But think of a baby who is learning how to walk. Oftentimes parents would hold them up by their hand, and it is a very similar process through which the baby learns to develop the movements, and also the brain is facilitating in this process. You may have heard of phantom limb, which is a condition that is experienced by most amputees, which is that they continue to experience the limbs or feel them, sense them, even though they've been amputated. This is what's known as a phantom limb. Now, some individuals, it's believe that about 20% are born with a missing limb. And these also report experiencing a phantom limb. And that is very interesting because these perceptions are very, very real. The existence of this phantom, that may be a hallucination for some, has actually been taken out of the context of myth and been supported by understanding the reorganization of the cerebral cortex with these individuals. Dr. Ramachandran, a neuropsychologist, was influential in understanding and helping us get a better sense of why phantom limbs exist. And it is interesting that Dr. Ramachandran benefited from research that was conducted in a previous time. In 1991, Pons and colleagues basically studied the sensory neurons by severing the arms of monkeys, and this is what led to the reorganization of the somatosensory cortex. This area that originally received input from a damaged arm now received input from areas of the body that were normally mapped to other parts of the somatosensory cortex. Based on this understanding, Dr. Ramachandran got an idea that perhaps phantom limbs were not in the stump or like in the limb itself, but in the brain. And perhaps the perception of a phantom arm originated from parts of the body that now were innervated to the original arm area of the somatosensory cortex. And this was actually in the late 90s. This hypothesis, that is known as the Ramachandran's hypothesis, basically was tested with one of Dr. Ramachandran's patients, Carlos. You may have read about this in chapter 10. And he basically asked Carlos if he would participate on a test. He basically touched various areas and parts of Carlos' body and then asked Carlos what he was feeling. Interestingly, when he touched the side of Carlos' face on the same side as his amputated arm, Carlos felt sensations from various parts of his phantom hand as well as his face. And this is what became known as a second map of his hand that was found on his shoulder. And you could see again how this part here would correspond to this area, and the two would correspond to this area, and that would have been the hand. Philip was another of his patients, and he also suffered from the severe chronic pain that sometimes is experienced in his phantom arm. For a decade, Philip's phantom arm had been frozen in like an awkward position, and so Philip suffered a lot of pain, specifically in his elbow. The question was whether Philip's pain would ever be relieved by teaching him to move his phantom arm. And so, knowing the important feedback on the movement, Dr. Ramachandran basically developed a special feedback instrument, an apparatus for Philip. This was basically a box that was divided into two by a vertical mirror. And Philip was basically told to put his good right hand into the box through the hole in front of the view and view it through the hole in the top. When he looked at his hand, he could see it in its mirror image. He was instructed to put his phantom limb in the box and to try to position as best as he could so that it corresponded to the mirror image of his good hand. After that, he was then asked to make this movement, synchronous movement, by basically making the symmetrical movement with his arms, his actual right arm and his phantom left arm, while viewing his good arm in its mirror image. And what Dr. Ramachandran did was basically send Philip home with a box and asked him to do it as frequently as possible. Within 3 weeks, Philip's pain had subsided completely, and this gave way to our understanding of how the phantom limb occurs. And as you can see, this is all based on the organization of the cerebral cortex and not the limb itself or the stump of the limb at all. I hope the understanding of how we've come about to discover our nervous system gives you a sense of hope as to the future of this field. Never before have we been in such a privileged position where we've been able to fuse a lot of disciplines, from engineering to computer technology and medicine and psychology, to understand individuals. And the hope of this fascinating field is that we will continue learning more and helping individuals who suffer from ailments to have a better life.